Kim, director of the UCLA Human Stem Cell and the Genome Engineering Center. And our center has two main functions. One is conducting independent research with active collaboration with your laboratory, potentially, and also operating the core facility for iPSC reprogramming and gene editing, led by Dr. Hyo-kyung Cha and Dr. Dennis Attar. With robotic automation, we recently established by supported by California government grant. And some participating institution will also access this service through the core-to-core partnership, which will be launched this year. So therefore, you can access our core function through your own institution. And this program is supported by the California Institute for Regenerative Medicine and led by the UCLA in partnership with Thermo Fisher Scientific. And this year we have nearly 200 participants from national and international institutions with a special topic in bioprinting and the AI-driven computational approach in stem cell research. And welcome, and please note that participants who attended 6 lectures will receive the certificate of the completion. And then now I would like to invite Dr. Catherine Platt from UCLA, following by Dr. Thomas.forbes and Dr. Omar Farah from Thermo Fisher Scientific. Dr. Catherine Platt, and could you introduce yourself and any remark?
Hello, everyone. Welcome to this online class. I'm very excited about it. I run a lab at UCLA on pluripotent stem cells and reprogramming to induce pluripotent stem cells. So I'm here to support Kitai's CRISPR and iPS core, and also support the Thermo team in any way I can throughout this class. If you have any questions, please email me. You find my email online. But really what I want to do is thank the Kitai's team in the core and the Thermo team for putting this together. I'm really grateful for everyone doing this. We have two amazing teachers, Omar and Tom. They have done it before with us, and the class has been wonderful. So I very much look forward to this, and I hope you guys will all enjoy this. So, but please, Omar and Tom, take it from here.
Sounds really good. Thanks, Catherine. Really appreciate it. And Omar and I'll speak for him as well. You'll hear him in a moment. But we're both very excited to do this course again. It was really well received the first time around, and it's a great opportunity, I think, for all of us to keep sharing the knowledge and the love for stem cells. So my name is Tom Forbes. I'm a staff scientist at Thermo Fisher Scientific. I've been with the company now for about 5.5 years, I'd say. I support our stem cells and neurobiology portfolio on the west coast of North America. My background and training are in developmental neuroscience. So I'll do some of the lectures here and there, but especially the neurobiology lecture that we'll be doing. And I'm based in Carlsbad, California. So really happy you guys are here, and looking forward to getting things kicked off today. Omar, I'll pass it over to you.
Perfect. Thank you, Tom and Kita and Catherine, for that really nice introduction. And just like everybody else, I'm super, super excited about the course. So I'm hoping everybody is. We have a great material lined up. And please feel free, like Kita and Catherine mentioned as well, to engage as much as possible, ask as many questions. There's literally no rules here, right? So whatever you guys want to know about, let us know, and we can see how we can help you out. I should mention as well my background too. I forgot that, but I finished my training in early embryo development and lineage specification, and then I moved to UCSD actually to finish my postdoc in placental research and sort of how the placenta invades the maternal bed and interacts with a whole bunch of different other cell types on the maternal side. And I joined Thermo Fisher about 7 years ago, and I've been supporting the stem cell neurobiology portfolios as well, but on the East Coast. So I'm Tom's East Coast counterpart.
Are there any other instructors on the course that would like to introduce themselves? And it's totally okay if not.
Yeah, I think they're coming later, Tom.
Okay, no worries, no problem at all. What we can do while I'm just going over a couple of the course objectives and the course structure, I thought it might be fun if everyone that's on, or those that would like to anyway, want to pop in the chat just their name, where they're from, and maybe a fun fact or something about themselves. So feel free to populate the chat while I kick us off with a little bit of a course overview. That would be great. But I'll get cracking on that while you guys pop into the chat. Again, thank you very, very much for joining us today. This will be the second edition of this course that we've done, this UCLA Thermo Fisher Scientific Stem Cell Basics course. What we're going to do today is just kick us off with an introductory lecture, which I'm going to begin with shortly. But before we do that, as I said, I'm going to spend just a couple of minutes talking about the course itself, some of our objectives in leading in the course, and again, just kind of getting you guys excited, hopefully, about the really good lineup I think we have, and the really cool and really interesting people that will be presenting as well, and sharing what honestly amounts to quite a vast knowledge of stem cell biology. So on the whole, really our overarching course objective is to provide foundational training for you on basic stem cell techniques. We've got all walks of life attending the course. We've got very, very new and fresh beginners. We've also got some experienced stem cell biologists as well. So to try and tick that box for everyone of making this an introductory and an interesting still course, we're going to cover topics like stem cell culture, reprogramming, gene editing, differentiation. And again, some of you may be at least partly familiar with this content, and some of you may not be familiar with any of it at all, and that's certainly okay. Again, the course is going to be led by faculty and staff from UCLA and Thermo Fisher Scientific for the most part. And then we have a couple of interesting guest faculty as well, which we're really excited about. We didn't have them last year, so it should be good. Again, the course represents, as Kitai alluded to, a shared resource activity component of the UCLA CIRM initiative, or California Institute for Regenerative Medicine, as we affectionately call it. CIRM is a state agency. It's established by the people of California, and the objective really with that was to help accelerate stem cell biology, regenerative medicine, gene therapy research, and so on and so forth. It gets a good amount of funding to help support the development of innovation, critical research, and also investing in the next generation of scientists, which is many of you, to hopefully lead the way and continue to carry the torch moving forward, and ultimately advancing regenerative medicine. So super excited that you guys are here. So we're going to talk about, as I said, many basics and some really sort of nitty-gritty concepts as well. We're going to kick us off today with some best practices for aseptic stem cell culture, which is obviously a really, really, really important backbone or foundation of successful research. We'll talk about human somatic cells, right? Things like blood or skin, for example, being converted or reprogrammed into stem cells, so the so-called induced pluripotent stem cells, or iPSCs. We're going to talk about some of the basics of stem cell culture techniques, expansion, maintenance, passaging of cells, and then along the way, we're going to give you some handy tips and tricks, which hopefully actually should help you put principles into practice in the lab as well. We're also going to touch on differentiation of a bunch of different cell types, so taking those blood or skin cells that I talked about, again, reprogramming them into iPSCs, and then ultimately differentiating those into specific cell types, think like neurons, cardiomyocytes, things like that. We'll also give you some really good insight, I hope, into the UCLA Stem Cell Engineering Core and some of the services that they offer in support of stem cell researchers. We'll dive into a bit of gene editing. We'll talk about clinical application of stem cells in regenerative medicine. And then I think the other big take home that Omar and I really, really want to emphasize this is the sense of community, right? Again, we've got some really, really excellent subject matter experts in the field, wide experiences, connections, knowledge, right? So I want you to leverage that, really, really. This is why you're here. You're here to leverage all of our collective experiences. And I think really use this course as an opportunity to enhance and expand not only your knowledge, but if you want, your professional network as well within the stem cell community. Now, just quickly, as far as content goes for each of the lectures, as you see here, the course structure is going to revolve around a series of 7, potentially 8 discussions or lectures spread out over the next couple of months. We're going to meet Mondays at 9 a.m. and for about an hour, hour and a half or so for each session. But we should have a bit more time at the end if we should need it. Again, the talks are going to be centered around specific topics: introductory stem cells, reprogramming, feeder-free culture, gene editing, differentiation. And again, we're really hoping to provide a comprehensive foundation, if you will, to stem cell culture. We'll also include some case studies and important literature as well. We'll talk a little bit about the history of stem cells and what's really become, I would say, an ever-evolving field. And then throughout, we'll also share some of the tools that we offer at Thermo Fisher at our disposal to help support stem cell research, as well as our collaborations with institutions like UCLA, who have a tremendous amount of resources and very talented people. Anyone have an idea what these are on the right here? Pop that into the chat if you do, what these cells might be.
Wondering, do people get an award if they're like an award, like a prize if they do? But let's see.
Yeah.
Lena already got it.
Yeah, so these are mouse embryos that were collected at what's called the blastocyst stage, so about 3.5, 4 days post-fertilization. You can see they're a tiny bit different looking from one another, but developmentally they're roughly at the same stage. And again, we'll get more into that kind of stuff shortly. This is just quickly an updated list of course instructors and their associated contact info. As I mentioned, I'm Tom Forbes, and I'm going to be leading the lecture today. But again, we're really, really happy and really, really fortunate to have all these experts on the call with us. And thank you as well for sharing a little bit about yourselves in the chat. That's super handy. It just gives us a quick idea of who you are, where you're from, and things that are interesting about you. But why don't I kick us off? Just lastly here, talking about course materials. We will always share their lecture slides after each of the subsequent lectures. They will be recorded as well, which is super handy. We'll share associated protocols, case studies. We'll try and share interesting links. Pretty much everything we share will be completely free. Some of it might have a little bit of Thermo Fisher branding on it. There's no way around that. But we're really not trying to push anything Thermo Fisher here either. This just happens to be led by us in collaboration with UCLA. We want you to get a lot out of this course, keep it very agnostic for your stem cell knowledge, and hopefully make it very, very useful for you. Again, we're going to share a lot of what we think has been cherry-picked as useful content. And again, the one thing I want to take home from this is really feel free to leverage your instructors as a resource as well. You can blurt out anything you want during the lectures. You can pop questions, scientific or non-scientific, in the chat. If we don't have a chance to answer anything, Omar and I or one of the other instructors will certainly follow up afterwards. We're happy to talk about anything science. We're happy to talk about career development advice. We can talk about our roles at Thermo, our roles in academia. We've all done quite a lot of things. And again, we're really excited to have you guys on this course. Really glad you're signed up. And please don't be shy here. Omar, anything else you'd like to add? Or do any of the instructors would like to add before I kick us off with the lecture?
No, I just I wanted to just reemphasize the point that you made, Tom, about like the educational piece, right? Of leaning in on resources that are already existing, so you don't have to reinvent the wheel. Something that comes to mind all the time when I kind of think about this is how we used to do, research back in our days. And I don't want to date anybody here, but for some of us, things like even PCR were very, very challenging in the beginning if you didn't have access to like a thermocycler and you had to set up water baths. And the reason I'm mentioning all of this is please feel free to ask questions and lean in on others that have done things like that in the past so that you don't have to make the same mistakes, or that you can kick off your work a little bit more seamlessly as well. But yeah, no, Tom, all yours. Please kick us off.
Sounds really good.
Also,
Go
just to
ahead.
briefly mention is that also this year, 3 special guests will be involved. And April 20, Dr. Ho will talk about clinical trial and the brain organoid. And also May 11th, and Dr. Sosa will talk about 3D bioprinting and then stem cell research. This is the new emerging area. And then also May 18, and Dr. Correa we'll talk about the integration of the system biology and the AI tool. AI tool in cooperation with the stem cell research is really, really hot area. So therefore, you can just have another opportunity to get some interaction. Thank you. Tom.
Perfect. Thanks, Kitai. Appreciate you mentioning that. Okay, so specifically as far as today, a couple slides on biosafety in the cell culture setting. I know this isn't biosafety anyway, everyone's favorite topic. It might be some of yours favorite topic, and that's fine, but this is super, super important information. It's always worth a quick mention at the start of any stem cell course. It's important information to have in the back of your mind, whether you're a beginner or an experienced user. We're going to then segue into some basics surrounding cell culture, and then we'll transition over to what will be just kind of a high-level overview of some of the science underlying the stem cell culture workflow that we support in our specific roles at Thermo Fisher. And then, as we'll often try to do, I thought it'd be interesting to round out the discussion today with a case study. I picked one on Parkinson's disease, go figure, a neurobiologist talking about something that's neurobiology related. But I think it's an interesting one. It's a really cool and clinically relevant story and incorporates some of the tools that we'll be talking about shortly. Okay, so biosafety, again, as good as any place to start any cell culture course is a quick mention on biosafety. Again, I'm sure a lot of you have heard a lot of this information before. You might even have required trainings at your specific institution, but again, it's worth reinforcing. You know, we've all had experiences in the lab, some of which were very good, and some of us have had some bad experiences as well in the lab, because I think, you know, we don't realize it when we're in the thick of our research and very, very focused on what we're doing, but even one small deviation away from biosafety in the lab can severely impact the health of yourself, of those around you, and it can also have massive consequences on some of the work that you've been doing and the outcome of your experiments as well. So a quick and gentle reminder on that from us, I think, is important. Obviously, a cell culture lab has a number of specific hazards associated with it. Much of it has to do with the handling and manipulating of things like cells and tissues, and even some of the toxic, corrosive, or even mutagenic solvents and reagents that we often use. So really the fundamental objective of a biosafety program is to reduce or eliminate exposure of the researchers to these potentially harmful biological agents, and obviously try and reinforce adherence as best we can to kind of standard, if you will, microbiological practices. And those standard microbiological practices are found in a document or publication called Biosafety in the Microbiological and Biomedical Laboratories. I'm happy to share a copy of that if anyone would like it, but it's an important document that gets updated quite regularly. As you see here, the document defines 4 ascending levels of containment. Those are referred to as biosafety levels, and we categorize those as 1 through 4. And again, basically these describe some of the practices and safeguards and associated levels of risks with handling particular agents. As I've highlighted on this slide here in blue, I'd say most cell culture labs are around a BSL-2 level. Obviously, the requirements can be slightly different depending on things like the cell line you're using and the type of specific work that's being conducted. And again, in the interest of time, we're not going to go through these too much, but I will call out BSL-2, which I think is appropriate and widely used for moderate risk agents that can cause human disease, often through things like ingestion or exposure through mucous membranes. Again, we're not going to go into the lab together this time around, but I do want to touch on some of the best practices to help create barriers, right, between yourself and some of those potentially hazardous agents. And again, we've always got to think as well, not just about the safety of ourselves, right, but also protecting your cell culture experiments from contamination by you, or maybe from contamination by some of your lab mates as well, who may not have such good lab etiquette. We always wear PPE, right? Personal protective equipment. That's what forms the immediate barrier between you and hazardous agents. This can be things like gloves, lab coats, gowns, safety goggles, right? Which you should always wear in the lab, right? Sometimes you need things like shoe covers, respirators, face shields for higher exposure experiments. But PPE is also used, right, in combination with other things like biosafety cabinets that help us contain those potentially harmful agents. So again, these are just standard guidelines for safe laboratory practice, but big take home here, always wear your PPE, change that PPE when it becomes contaminated, right? And then always, always, always report incidents. Doesn't matter if they're big or small, to the appropriate personnel at your respective institution. We're always looking to make this safer and more effective. Now, the specific requirements of a given lab can vary depending, as I said, on the type of research that's being conducted. But pretty much all cell culture labs have the common requirement of being free from pathogenic mechanisms, right? And they typically then share some of the same basic equipment that we know is essential for culturing cells, right? Things like cell culture hoods that allow us to have an aseptic work area. They also help us contain things like infectious splashes or even aerosols that are generated by a lot of the procedures that we do. On the right here, we've got a fellow standing at an incubator, right, which helps us maintain things like temperature, CO2 levels, humidity, and so on and so forth, really giving us an appropriate environment for cell growth, right? And in many cases, one of the biggest goals of these incubators is not just right, but also mimicking the conditions in the body where perhaps those cells that you're working with originated. Now, again, other pieces of commonly seen lab equipment, we've got microscopes, hemocytometers for things like imaging and counting cells respectively, refrigerators, water baths, centrifuges, media aspirators, cryo storage, sharps containers is a big one, and biohazard containers. Again, these are always labeled red, colored red, and they must be labeled appropriately. And again, I think most of you have at least some experience in a cell culture or research setting now, but one day you might be running your own lab. And pioneering stem cell research. So this is just a quick reminder of a few of those things. And then lastly here, sterile technique, right? That's the only other thing I want to drive home on this. It's a really, really important component of stem cell culture. We want to obviously protect your cells from contamination by you. And then on a larger scale, right, more importantly, protect you, the researcher, from exposure to many known and hidden lab risks. So this slide is just a gentle reminder to work thoroughly, stay organized in the hood, maintain a sort of clean to dirty flow, as you see here in this schematic here. Always decontaminate your work surfaces before and after each use. Clean up immediately after spills. Have a regular cleaning schedule for the lab equipment that you're using, whether you're using it that day or that week or that month or not. It needs to be clean when the next person comes around to use it. Remember too, good lab etiquette. I'm sure we've all got nightmare stories here of folks that we've shared spaces with in the lab. There's a lot of common real estate in a cell culture laboratory, so you have to be considerate of others too, and obviously try not to ruin anyone else's work by being careless. Again, a lot of this is common sense for most people, but it is, I think, worth a gentle reminder. It helps us reduce the risk of exposure to these potential hazards, and ultimately helps us ensure that cell culture experiments are going to be successful. Any questions based on any of that so far? Okay, I'll keep us going then. I'm going to switch gears a bit here and start diving into some of the science underlying cell culture basics, right? Again, some of this content might be a bit of a review for some of you. It might not be either, and that's okay. But it is important, I think, to sort of set the stage and touch on a couple of fundamentals that are important to understanding some of the concepts that we'll be talking about together in the course. So what exactly are stem cells, or cells, I should say? Let's start with cells, right? Cells are the smallest unit of life, whether that's a whole organism, bacteria or yeast or some types of parasites, right? Or cells that are integrated into parts of a larger entity, right? Like think in particular tissue or a particular organ or even an organ system, right? Our cardiovascular system, for example. But cells are the basic structural and functional unit of every living self-replicating organism, right? And there's really two main classes of cells, eukaryotes and prokaryotes. Prokaryotes include things like bacteria, archaea, cyanobacteria, those blue-green algae, which are super cool. And the term eukaryotes includes things like animals, plants, fungi, insects, humans, right? We're composed entirely of eukaryotic cells. Most scientists believe that eukaryotes are evolved from prokaryotes, which is super interesting, about 2.7 billion years or so ago. Really with the primary distinction between them being that eukaryotic cells have a membrane-bound nucleus, and prokaryotic cells do not. The nucleus, as I'm sure many of you know, is where eukaryotes store their genetic information. But although these two types of cells, eukaryotes and prokaryotes, do have a few differences, right? They do share some common features as well. DNA, right, which is the coding material that determines all the characteristics of living things. DNA in a cell is found in a particular area or individual pieces known as chromosomes, right? So both of these cell types have chromosomes, although eukaryotic cells have many chromosomes, which undergo things called mitosis and meiosis during cell division, while most prokaryotic cells consist of just one circular chromosome. Both also have a plasma membrane, right, which is what separates it from the surrounding environment. Kind of acts as a selective barrier, if you will, right, for incoming and outgoing materials. Now, does anyone remember what that cell membrane is often made of? Might be digging deep in the knowledge bank there for some of you, but what is the cell membrane made of? Omar, feel free to shout out. I can't see the chat, so
I
feel
was
free
gonna
to
say, I think Say already got it. They're saying lipids, lipids, phospholipids, lipids, which sounds like a song, which I love, but they've
Yeah,
already gotten it. Yeah, phospholipids.
good. What about the charge? Do we know or remember if it has an overall net positive or net negative charge?
They're coming in again. Negative, negative, negative.
Good.
Yeah.
Good. Yeah, so that barrier, if you will, is comprised mostly of phospholipids, right? And it has a net negative charge. We've also got the cytoplasm, right, in here, right? It's that kind of like jelly-like fluid within a cell. It's made up of lots of water, salts, proteins, right? Does anyone remember the name of the organelles that make proteins? Yeah, ribosomes, right? And these proteins are everywhere. They can be in the outer membrane, embedded in there. They can also be secreted to affect other cells, right, through different factors and cell signaling. How do cells get their nutrients and energy, though? Any ideas on that? The cells from tissues are normally supplied with nutrients through blood, right? But in cell culture, this is where media comes in, right? Cell culture media. And that cell culture media allows us to replace a lot of the things that are normally supplied by, say, the bloodstream, needed for things like cellular metabolism. Now, remember, in vitro cell culture is a method that we use, or a label that we use for studying the behavior of animal cells, right? Kind of mimics the setting of a living organism, if you will, and allows us to control for variations, or as many variations as we can as possible, to sort of recapitulate the natural environment of those cells, right? So if we think about that, even at, let's say, the most basic level, when we use cell culture media given to the cells, we want cell culture media that's appropriate, right? That provides the specific nutrients, that provides a stable environment for those cells in which to grow, right? And we'll talk about some of those specifics later on in different types of media, and we'll give some examples down the road. But we have to account for this in cell culture media, things like salt balance, sugar levels, right? Vitamins, amino acids, pH, temperature control, so on and so forth. There's also compensation for different attachment proteins and all the different growth factors that we use, depending on the specific cell type that we're using. So media choice and all the factors that go into that and creating this nice environment for your cells, right? They're important considerations. And again, we'll dive into more of those specifics later on. But what about stem cells, right? Our beloved stem cells. What are PSCs, or pluripotent stem cells? Well, these are, I would say, pretty remarkable cells. As we've already alluded to, these are quite a hot topic in science and regenerative medicine at the moment. But these stem cells, originate from early stage embryos. And you can think of them as having two defining characteristics, right? First is the ability to self-renew indefinitely, right? And also the capability then to differentiate into varied cell types from the three so-called germ layers, right? Endoderm, mesoderm, and ectoderm. What that means is they can give rise to just about any cell type in the body. So how on earth does this happen, right? Well, as you see here in this schematic on the left, shortly after fertilization, something called the zygote starts to divide, right? 2 cells turn into 4, 4 cells turn into 8, and so on and so forth, until you get to what's called the morula stage at the top here. And this is where we actually start to see a lot of the physical changes happening, right? And so this cluster of rapidly evolving cells starts to almost organize itself, right? Migrating, compartmentalizing, and in a lot of ways, sort of beginning to adopt their cell fate, right? And it's here that we reach what's called the blastocyst stage. And the word blastocyst refers to the stage that the human embryo reaches about 5 to 6 days or so after fertilization. And the big take home here that I want you to think of is an early blastocyst, right, is comprised of 2 different cell types. We've got an outer layer of cells called the trophectoderm, which gives rise to extra embryonic tissues, right, like the placenta and the amniotic sac. as well as a group of cells located on the inside, right, which are appropriately named the ICM, or inner cell mass. And so the ICM, the inner cell mass, are the ones that give rise to the embryo proper, or the fetus, right, as pregnancy progresses. So pluripotent embryonic stem cells originate from these inner cells within a blastocyst, and these stem cells can become just about any tissue in the body, excluding the placenta. So think of the inner cell mass as what gives rise to those three germ layers, right? Endoderm, mesoderm, and ectoderm. And that's where the name stem cells come from. Those embryonic cells of the inner cell mass, which again, by definition, are self-replicating, meaning that they have the capacity to divide, right? And that they could potentially give rise to themselves again, as we see here in the middle, right? In this self-renewal box. And again, call me biased if you will here, but I think stem cell biology is super interesting. And, over the last 6 decades or so, really has become one of the most exciting and I would say rapidly changing areas of scientific investigation. I think I'm probably talking to the right crowd here when I say that, but we have so much potential in stem cell biology and regenerative medicine for providing what are often, or I would say very soon, going to be a lot of curative approaches for a lot of different disorders that originate at the stem cell level. So really, a lot of the power underlying pluripotent stem cells especially lies in their regenerative potential, right? And all the different applications in which we can use them. In basic biology, we can use pluripotent stem cells to help us understand why things like early human development and cell differentiation processes occur. We use stem cells in disease modeling, right? They allow us to create really, really accurate depictions of human disease. Turn that enables better understanding of lots of different disorders that affect humans. We can use them for things like the screening of new pharmaceuticals. And then in cell therapy, right, which again is a very, very hot topic at the moment, pluripotent stem cells especially, I think, hold so much promise in generating healthy cells to do things like replace damaged tissue, maybe even offering as a cure one day for different debilitating diseases. And again, we're going to unpack some of those clinically relevant applications throughout the course and how, again, keep driving home how stem cells really are the future of science and medicine. Now,
Hey,
I think,
Tom.
go for it, Omar. Yeah, please.
Maybe just a quick mention on the cell therapy side, because this is
Please.
rather recent and interesting. The first two stem cell derived cell therapy applications were recently, or treatments were recently approved by the regulatory bodies in Japan. One for Parkinson's and another one for cardiac disease as well. And I believe these are the first two widely approved PSC therapies, which, to Tom's point, right, has been the goal of what we're trying to do here, right, is to figure out a way to harness this regenerative power of these cells and use them in some sort of way as a repair kit for current disease and pathology. And that's already happening now, right? Like at the beginning of this year was the approval for those two therapies. So super exciting time. And I think, correct me if I'm wrong, Tom, but I think we're going to talk about both cardiac and neural indications as well throughout. So you'll hear more and more about this as we kind of unpack the course throughout the next several weeks.
Yep, absolutely. Those are two great examples, and I think two really clinically relevant examples as well. And we're going to be talking about those in both 2D and 3D with some really clinically relevant and nice data as well. One thing I want to get back to with regards to just introducing the notion of stem cells is that not all stem cells are created equal. That's important, right? If we go back to our original definition with the two so-called rules, right, of what it takes to be a stem cell, the first was that they're self-replicating, right? And this is true for all stem cells. But the second part of that definition is their ability to give rise to other cell types. And so this differentiation, if you will, is where some stem cells are in fact not equal to others. So there's stem cells that can differentiate into almost anything, right? And then there's stem cells that are much more restricted that can only give rise to certain cell types. And so this is where we use certain nomenclature. You may have seen some of this before, whereby if I say, for example, a stem cell is totipotent, that means it can give rise to everything, right? Not just the embryo, but those extra embryonic tissues as well, including the placenta and the amniotic sac. Sort of one notch down from that, if you will, from that all-encompassing totipotent capability, we've got stem cells that are pluripotent, right? And this is a word that we throw around quite often. These are cell types that can give rise to most cell types in the body, right? In other words, those that derive from the endoderm, ectoderm, and the mesoderm, as you see here on this slide here, but not those extra embryonic tissues. And then finally, the last of the nomenclature that I want to mention is multipotent stem cells. And these are much more limited in that they can only give rise to a subset or specific subset of cells within the endoderm, within the mesoderm, or within the ectoderm lineage. So a multipotent stem cell, for example, that's an endoderm progenitor, can only give rise to endoderm lineages, but not the ectoderm or the mesoderm. So the big take home here, not all stem cells are created equal. Now, another nomenclature that we like to use or classification for stem cells is how they're derived, right? And we've got loads of modern technology at our fingertips, right, that allows us to sort of further categorize these different types of stem cells. So as we kind of alluded to or talked about, embryonic or fetal cells, those are derived from fetal tissue or embryos specifically. Again, here we go to that inner cell metabolism. So taking those big nucleated cells at the top here, right? Growing them in culture. These cells are therefore pluripotent. They can differentiate into almost any cell type in the body. They can also renew themselves indefinitely. Next in the middle here, right, we have induced pluripotent stem cells, or iPSCs. We're going to spend most of our time in this course talking about these reprogrammed cells that are also pluripotent, right, as the name implies, and that can also renew themselves indefinitely. And then lastly, at the bottom here, we've got adult stem cells. And these are those that are isolated from tissue, organs, or blood, right? And these, again, a bit more specific. They can give rise to only related cell types, like hematopoietic, neuronal, mesenchymal stem cells as specific examples. And these don't renew themselves indefinitely, as the others do. Now, while we're on the topic of stem cell derivation, we like to every now and again put some pretty pictures in here. I think that helps keep it interesting and relevant. I'm a very visual learner, so we'll often have nice pictures throughout. But this is just an example of how we can dissect those embryonic cells from that inner cell mass. We then plate them onto a dish, as you see here in this image, right? And again, we call these embryonic stem cells because they're derived from an embryo. And again, these cells are pluripotent, and they can differentiate into almost any cell type in the body. We can also obtain embryonic stem cells by a really, really interesting process that involves removal of a single cell from that blastomere that I talked about. So what happens in this case is the egg is fertilized by a sperm. It grows, begins to divide, as you see across the bottom here. And then right around the 8-cell stage of the embryo, we can actually separate those cells, take one cell out. This is called our blastomere, right? We can then nowadays culture that blastomere in the lab, which is then able to give rise to the entire embryo again. So the take home here is that with just one cell in this instance, you can have a colony that starts to form and differentiate, ultimately then giving you another stem cell line.
And Tom, sorry, before you move on, one other quick
Please.
thing that I'll add here.
Yeah.
Just a quick the slide before, just a quick reference to the blastomeres and the pluripotency status, right, of or the potency status of the stem cell that Tom mentioned. So you'll notice here that if you remove a single blastomere from an embryo, from let's say an 8-cell embryo, if you separate that blastomere into 8 separate cultures, that single blastomere, right, has the capability of remaking that entire embryo again, right? And at that stage, that's because those cells are totipotent, right? They can give rise to everything. But later on, if you try to take a single cell from, a later developed embryo, it won't have that same capacity because it's already started somewhat down this like lineage specification. And at the blastocyst stage, right, as Tom mentioned, you have the inner cell mass, and then you have the trophectoderm, the two different types of cells that have been specified, right? So just keep that in mind, like about where the cells are on that spectrum of quote unquote stemness as well. That gives them a whole bunch of properties too.
Thanks, Omar. Okay, now that we've introduced a couple of different types of stem cells, I do want to touch just briefly on some of the history underlying stem cells, because I think this is really interesting as well. And I'll start with a question, and that is, how long do you think the concept of stem cells, how long has that been around, right? How long have stem cells been around? You can blurt it out, you can pop it in the chat. While you're answering, I'll say this is one of Omar's favorite random questions to ask someone in a bar as well. It usually involves a few funny looks, and then he tells them he's a stem cell biologist and often ends up buying them a pint of Guinness. But any guesses? Omar, can you read the chat? Anyone close?
So people are saying 1970s, 70s, 40 years ago. So we have some guesses.
Okay,
Anything else?
those are good, but with all due respect, way off. So
Yeah.
the concept, as we know it, of stem cells and stem cell properties dates back to the late 1870s. And a German scientist, a man by the name of Ernst Haeckel, right? And he used the term Stammzell, Stamzelle, to describe what he thought was a fertilized egg and sort of a structure, if you will, that was the origin of all other cell types within an organism. So that's an awful long time ago. And obviously, there's been loads and loads of work done since then, but there's been a few big milestones along the way in stem cell biology. And the next big one I want to talk about just quickly today was in 1953. 1953 was a pretty important year in science. You may have remembered this date for some other reason, and that's because it's the year that Watson and Crick discovered DNA's double helix. But it was also the same year, 1953, that two Canadian researchers, Clermont and Leblond, they published a seminal paper that basically identified the theory of stem cell renewal. They discovered a population of cells that they knew were responsible for the continuous production of sperm in rodents. So super, super interesting. So the big take home here is stem cells have been around for a long time. I think, in fact, longer than most of us anticipated or expected. We've obviously had big things and big milestones happen along the way, right? We also had a lot of public recognition, if you will, with Dolly the sheep in the late 90s. I believe it was around 1996. Dolly, as you see here, she was part of an experiment in Edinburgh, Scotland, at an institution called the Rosalind Institute. What they were doing was evaluating different techniques to produce genetically modified livestock. And so what they did is they took a nucleus from one cell, they put it into the cytoplasm of another, and then they put that clone cell into a surrogate mother. And what they did next is evaluate what or who that baby resembled, right? Did it resemble the original sheep that they took the cells from, or did it resemble the surrogate? You can see here that Dolly has a white face, right? And that was different to the surrogate mother, who had a much darker coat, darker face color. That really allowed them to demonstrate then that Dolly was not the offspring of the surrogate, but of the donor cell that they had used. Another example was in 1998. Scientists discovered how to take cells from an embryo, grow them in vitro, right, in a lab setting. And then maybe the biggest, or I would say probably arguably the most important of all those discoveries was in 2006. And we'll talk about this throughout the course here and there, but this is when Shinya Yamanaka and Jamie Thompson really, I would say, catapulted stem cell biology and stem cell research by introducing, if you will, an entirely new way to reprogram cells, right? Adult cells, all the way back to a pluripotent state. An amazing concept. And again, we'll talk about that shortly when we talk about reprogramming. But this pioneering work led to, I would say rightfully so, a Nobel Prize for Yamanaka in 2012. Now, in today's modern medicine world and modern society, right, again, stem cells hold an awful lot of potential. They're also, really a hot topic financially as well. There's so many different areas where stem cells can have an impact, right? Regenerative medicine, drug discovery, developmental biology, as I alluded to here, right? With the recurring theme, if you will, of stem cells allowing us to recapitulate the human state through all these different relevant models, right? So we harness their power to then understand and treat a whole bunch of different human disorders, all in a physiologically relevant manner. And again, money often rules the world, unfortunately, but just to put a price tag on some of the impact that stem cells are having and their potential, right? We know the global market for stem cells is about $20 billion or so at the moment, an awful lot of money, and that's expected to continue to grow quite rapidly. So again, stem cell biology is a really exciting and good field to be working in. It's rapidly expanding, so you're all in a good space. So along those lines, what I thought we'd do sort of here on out until we get to the case study at the end is take a just kind of high-level overview look at the pluripotent stem cell workflow. And again, we're going to deep dive into each of these different pillars or buckets of the stem cell workflow over the next couple of months. But today, what I'm going to try and do is just dip our toes into each of these buckets, if you will. And sort of to set the stage and hopefully tell what we hope will be a comprehensive story with content building on itself, and I'll hopefully put things into context as the course progresses. So let's dive into that workflow just a little bit here. And I think this is a slide I use all the time in a lot of the talks that I give for Thermo Fisher, because I think it's important to call out stem cells as models, right? And really the utility, if you will, of any viable cell model that you're using is its capacity to ultimately predict outputs that translate to organisms, right? So if you're using a model, any kind of model, it has to be physiologically relevant, right? Otherwise, it's probably not worth using. And this is hard to do, right? We know that humans in the middle here, they're an orchestra of about 37 trillion cells or so. Sometimes the orchestra plays well together. Sometimes the orchestra doesn't play well together. And we know that sort of underlying all that is the genetic code, right? It sort of dictates or substantiates the structure of our body, how it functions, right? How things integrate or they disintegrate over time. And I will say, understanding and ultimately connecting how all these different inputs and how all these different outputs align is very challenging, right? Let alone the fact that we can't, say, ethically or practically experiment on human beings outside of, say, the strict guidelines of clinical trials. But in modern medicine and modern stem cell biology, there are a lot of outputs and tools that we can measure, right? We've got symptoms, we've got biomarkers, we've got morbidity, mortality outcomes, and so on and so forth. And then from those different outputs, we generate our models, right? And that allows us to even further decipher things like gene expression, protein function, cell phenotypes, the impact of media systems, which we talked on earlier. The big take home here that I always want you to think about, though, with cell models is all of these are very, very much a reciprocal system, right? And as selfish as we sound, right, we strive to relate all of these results back to the human being. Now, along those lines, right, one of the keys to successful cell culture is creating the most realistic environment that you can, right? But we have to do that in a way that the cells or whatever we're working with can still be analyzed. And so I would say, as I've alluded to here, in the grand scheme of things, we have at our disposal a number of different model systems for human disorders. I've just listed a couple of them here. This is obviously not an inclusive list. Starting on the list here, we've got planar or 2D culture models. These are widely used, they're readily available, they're quite easy to manipulate. But if you think about 2D or planar cultures, right, they often lack a lot of the particular cellular architecture and cellular diversity that we need to closely mimic human disorders, right? And as a neurobiologist, I would say this is especially true in areas of the body like the brain. We also often employ behavioral studies, right? And these are nowadays often performed in rodents. And I will say they're a nice bridge between, say, the inner workings of a particular organ and what we see for functional output. But you've always got to remember with behavioral models that they're typically not directly translational, right? Because we're often dealing with animals after all. And we'll talk about this a little bit more in the course as we progress, but there's a big initiative of the moment for something called NAMs, or new alternative methodologies, that's trying to steer science a little bit away from some of these animal models and getting more into sort of cellular 2D and 3D models that we know do also recapitulate a lot of the things that you'd see in human beings. Now, the other thing to keep in mind with a lot of animal studies and a lot of the things that we use as tools is that they're often not directly translational, right? And a lot of that has to do as well with adult onset disorders that we might study. And again, in the concept of the brain, think about neurodegenerative disorders like Parkinson's disease, right? It's hard to study something that impacts our physiology so late in life before things like behavioral changes can even be detected. Again, so there are limitations to these models, which again I think is another reason to start getting away from them. Now, we also use human tissue sometimes. Human tissue is great. It gives us a nice intact structure with which to work. That intact structure then is kind of a 3D microenvironment. But often when we're using human tissue, right, it lacks that functionality component because, as you might imagine, we're typically not performing experiments in live tissue. Not to mention that there's always limited availability, right, with the amount of, say, post-mortem and cadaver tissue that we can actually get our hands on as researchers. And then lastly on the right here, 3D models. And there's lots of different types of 3D models, I know, but they're great. This is a really, really cool space. I think one that's very interesting to me in the stem cell biology field. 3D models are often derivable from human cells. That means they do often closely recapitulate things like cytoarchitecture, inherent functionality of, say, a particular organ. And so generally speaking, 3D cultures do replicate quite well a lot of the in vivo conditions that we would see, especially say compared to some of the 2D systems that we use. And cells cultured in 3D typically provide data that are therefore more physiologically relevant, right? So closer to that of cells in their natural environment. Now, we'll talk about this throughout the course, but within the stem cell space, there's really two types of 3D models that we work with at Thermo Fisher, and those are suspension culture and organoids. And again, we'll unpack those a little bit more down the road and give you some nice clinically relevant examples of each.
Hey, Tom.
Yes, Omar.
Do we ask the group if they think one is better than the other? I'm interested to hear their
Please.
thoughts on this. So maybe just in the chat, does anybody think that there is a model like out of these four that is better than the others? Let's see what folks say. So you have 3D models. All models are wrong. Some are useful. It's a good point, Jane.
Yeah.
3D. So this is a good point. This is what I wanted to ask this question about, right? You guys are technically right in the sense of 3D models may be more physiologically relevant in certain cases, right? But a very important point that I want to make here is there are advantages and disadvantages to each type of these models, right? So depending on what question, your biological question you want to ask, you may have to shift a little bit in your approach, right? If you want to control variables a little bit more, then using a 2D culture or planar culture, it may be an interesting avenue because you can control variables there a little bit better. If you are looking for more physiological relevancy on function or maturity, then perhaps you want to go on the 3D route. I would advise not to look at them super in that like wider like black or white kind of lens, but think about it again as a spectrum of where do you place your chip depending on what you want to accomplish, right?
Really great point, Omar. Thank you. Yeah, and there's no wrong answer here on necessarily the type of model that you're using, right? Different strokes for different folks, and you know, you'll hear Omar say this, I'm sure, at least once, but stem cell researchers are a pretty stubborn group. Often they have a model system or a protocol that they know works, and they'll often stick with that, and that's perfectly okay. But you know, the point that we want to drive home in this course as well is there are other avenues, there are other types of models in the grand scheme of things. So it's always important to consider, am I using the best model for my particular line of research and the big picture question that I'm trying to answer with the research that I'm doing?
Yeah, can I mention something?
Please, Kitaya.
Yeah. So also as Omar said, and then there is sometimes timing issue is also very important. And then what type of the cell type is generating, because it's very hard to control because everything is mixture of the cell. That is the reason why another emerging technology is bioprinting. We will isolate a specific cell type and then three-dimensional manners, and we arrange each cell type to mimic the exact in vivo condition. And then we are going to discuss this in the one of the guest speakers.
Great point.
Yeah, excellent. All right, so let's talk about actually getting cells for in vitro modeling, right? And I would say, historically speaking, this has not at all been an easy thing to do. But again, big picture, with the advent of iPSC, induced pluripotent stem cell technology, we've really taken some big leaps and bounds here in creating models that correlate well to human disease. And so on the left here is kind of this notion, if you will, that we could take a donor, and from that donor, let's say me, I can take blood or skin cells, for example, and then through the forced expression of different protein transcription factors, I can actually reprogram those cells that I took into induced stem cells, which we'll talk about in a minute. We can then edit those cells if we want to introduce, let's say, a mutation. We can revert that mutation if we want to. And all of that allows us to really home in on, if you will, the contributions of even single specific genes or groups of genes with respect to a particular disease. And again, towards the end of this talk, I'm going to talk about that a little bit in a 2D setting and in the context of Parkinson's disease. Now, as you see on the right, skin and whole blood are obviously common sources for iPSC, induced pluripotent stem cell generation. And I've just listed here a couple of important considerations in actually obtaining those cells for modeling: patient comfort, sample size, cell number desired, right? Upfront manipulation requirements, the cost, right? The desired reprogramming method, and obviously the intended downstream application. And again, we'll touch on some of this stuff and unpack it throughout the course. Once we actually have those cells for in vitro modeling, right, the next bucket in our workflow that I've been talking about or alluding to is that process of reverting a somatic or adult cell back to a pluripotent state such that it behaves like a stem cell again, right? As we mentioned, that became really famous in 2006 when Shinya Yamanaka and his group were actually able, for the first time, to turn those somatic cells into iPSCs by forcing the expression of 4 different transcription factors, right? We had OCT4, SOX2, KLF4, and c-MYC. And again, he won a Nobel Prize for this work in 2012. Now, there's obviously multiple ways, as you can see here on this slide, to deliver those factors into cells to start the process of reprogramming. And so, and this again is not a comprehensive list by any means. So how on earth do you pick which reprogramming method to use? Again, as I mentioned on the last slide, I think you've got to consider the safety of the method, right? Things like the consistency of the system, the workload of the reprogramming method, even the cell type of reprogramming, right? And again, we're not going to cover everything on this slide today, but from a bird's eye view, I wanted to highlight that you can really deliver these factors through 2 main ways. We've got integrative and non-integrative methods, right? As we see here in this red box, I would say more traditional technologies, think things like lentiviral delivery that integrate into the genome of target cells, right? And what that means is the resulting iPSCs and then cells differentiated from those iPSCs then contain foreign DNA. And that can, of course, be problematic for use in things like cell therapy or even drug discovery applications. Nowadays, though, we often employ these non-integrating methods, as you see here in these green boxes. And these allow us to generate iPSCs that don't contain detectable vectors or transgenes, the most efficient of which, or at least very highly efficient of which, are sendivirus particles and episomal DNA vectors, which I've circled here in red, both of which we use at Thermo Fisher, and both of which support the reprogramming of a wide range of somatic cells. And Omar is going to deep dive into reprogramming in one of our later lectures. Again, switching gears here slightly and continuing on within our stem cell workflow overview, the next bucket I want to dip our toes into is genome editing. I think genome editing is absolutely fascinating as well. This is such a cool use of modern technology and basic stem cell biology sort of all bundled into one. But the basic approach underlying genome editing, right, is one in which we're adding, replacing, or removing DNA bases, right? That said, I think it's really important to emphasize that the genome is relatively resistant to change, right? DNA in our bodies is very important, right? DNA is responsible not only for encoding all the necessary functions within a cell, right, but also determining things like individual differences, right, between cells or between people. So really what I'm getting at is if DNA was really easily altered, a lot of essential cell functions could be very easily disrupted, right? Often, as you might imagine, in undesirable ways. And so really to protect against those inadvertent changes, cells have developed over time these inherent mechanisms, if you will, to ultimately proofread and repair their genetic code. And what's really cool about gene editing is we actually tap into those when we're using these techniques. So when we use genome editing to introduce all kinds of desired phenotypic traits into cells or even organisms, right, what that does is allow us to create even better models, right? And again, there's a wide array of applications for those models. I've listed a couple of them here: functional genomics, drug discovery, epigenetics, cancer research, right, just to name a few. Now, again, we won't jump into too much detail on this. We are going to talk about genome editing in a later lecture as well, but just a couple of basics, right? When we talk about gene editing, we often talk about designer engineered nucleases, right? And those include things, buzzwords that you might have heard like CRISPR or TALENs, right? That are ultimately designed to target nucleases to specific sites in the genome, allowing us then to create double-stranded breaks at specific or desired locations, as you see here with this top red arrow. Following those double-stranded breaks, what happens is those natural repair mechanisms of the cell that I talked about actually begin to heal that break. And this is typically done by one of 2 mechanisms. And again, we won't go into too much detail here: non-homologous end joining and homology-directed repair, as you see here in the middle. And again, we'll unpack some of that more later on. So big picture, genome editing is now routinely achieved through technologies like CRISPR and TALENs, right? Again, as I said, both of which program target nucleases to very specific sites in the genome, creating double-stranded breaks. I'd say maybe the most popular and arguably the most sort of name-wise, namesake recognizable of these engineered nuclease systems is the CRISPR-Cas9 system over here on the left. CRISPR stands for Clustered Regularly Interspaced Palindromic Repeats. Absolute mouthful. There's no need to remember that. But this is an RNA-guided DNA nuclease that was, I would say, quite interestingly, originally identified as part of the bacterial immune response pathway. In bacteria, these are actually part of their adaptive immune system that helps protect that bacterial organism against invading DNA. What scientists did, though, and sort of figured out, is they gained an understanding of this molecular pathway, and then they co-opted it, if you will, to be used for eukaryotic engineering. What we need for CRISPR-Cas9 is an RNA-guided DNA nuclease system, so bacterial enzymes that allow us to cleave that double-stranded DNA. That nuclease or enzymatic activity is conducted by a protein, and that protein is called Cas9. The way I was taught this was to think of Cas9 as a pair of molecular scissors, if you will, that allows you to cut those two strands of DNA at specific locations in the genome, and therefore enables bits of DNA to be added or removed. We also have a piece of RNA called a guide RNA or gRNA, and that guide RNA is designed to find and then ultimately bind to a specific sequence in the DNA. The guide RNA has bases that, as you might imagine, are complementary to those of the target DNA sequence. What that means then is that, at least in theory, that guide RNA will only bind to the target sequence and no other regions of the genome. And so this guide RNA or gRNA consists of a really small piece of a pre-designed RNA sequence, typically about 20 bases long or so, and it guides or recruits the Cas9, those molecular scissors, to the right part of the genome based on that sequence, making sure that that Cas9 enzyme cuts again across both strands of that DNA at exactly the right spot. So the big take home for CRISPR-Cas9 is that it's a novel gene editing system. The system consists of two parts, Cas9 enzyme and the guide RNA. And again, that allows us to modify, edit, delete, or even correct specific areas of our DNA by capitalizing on an endogenous repair mechanism. The other gene editing system to touch on quickly is called TALENs, as we see over here on the right. TALEN short for transcription activator-like effector nucleases. Again, no need to remember that. Just think of them as TALENs. These are also based on effector proteins, and again, these are also secreted by certain types of bacteria. In this case, though, bacteria that we know rewires host transcription and are actually known to cause major diseases in crops and plants. So these TAL nucleases or proteins contain amino acid repeating motifs, this time though with variable positions. This is really cool and really useful because it allows us to recognize specific DNA nucleotide sequences with sort of an unrestricted targeting of any sequence in the genome. And a big part of why they can do that anywhere in the genome is because they're a bit more general. They don't have some of the same constraints that CRISPRs do. The CRISPR systems do something called a PAM motif constraint. Now, TALENs, just for a quick comparison, are, I would say, limited to more simple mutations, unlike CRISPR. CRISPR allows us to introduce multiple gene mutations if we want, all concurrently with a single injection. CRISPR transfections also do tend to have a slightly higher efficiency. And that's not to say they're necessarily better for certain applications, but I would say CRISPR is probably a little bit more widely used systems. TALEN also often results in something called a mosaicism, in which we know that in many cases that mutant allele is present in only some of the cells that are transfected. So just a little comparison between the two, just a brief introduction of each. And again, we'll talk about these in a bit more detail later on in the course. Now, another important thing to touch on with regards to workflow considerations is the characterization of your cells, right? And this is something that's really, really important that unfortunately I think gets swept under the rug by some researchers, but it's a necessary step at certain intervals throughout the workflow. What stem cell characterization allows us to do is basically identify and analyze some of those fundamental properties of stem cells, right? Think their identity, their potency, their functionality, right? So characterization of your cells is really, really important as we move through those steps of the stem cell workflow. We would say we typically recommend performing evaluation of factors like genomic analysis, pluripotency, and sterility at kind of any major junction in your workflow. And again, as Omar alluded to, this is especially important in clinical settings, right? As we see here in this cell therapy workflow, right? And again, as I alluded to in the very beginning of this talk, what these types of things allow us to do is ensure safety and also factors like efficacy, right? Confirming stem cell identity and functionality is super important because it allows us to assess the health of the stem cell cultures, many times even detect changes or contamination that can affect those stem cell properties, right? And at the end of the day, ultimately determine their suitability for use, right? Characterization also allows us to determine how stem cell lines, because there's a lot of them, those stem cell lines behave, right? What's their ability to self-renew? What's their ability to differentiate, right? How do they respond to different signals, right? The other big take home here for stem cell characterization is if you think big picture, right, it also allows us to make sure that our results are reproducible, comparable, right, across different laboratories and different studies, which in this day and age is super important. Just to put that tiny bit into context here, there's an awful lot of words on this slide, I know, but I just want to emphasize the importance of characterizing your PSC lines. We know that about 20% of PSCs can accumulate chromosomal abnormalities, and so making sure that your starting material and the material that you're working with throughout the workflow is in its best condition is super important, right? Again, especially with regards to ensuring things like the consistency and the reproducibility of data. That's super important when we're working with stem cells, and we want to make sure that we're doing that. We want to reduce variability, and we want to increase safety, right? Because honestly speaking, it costs you a lot of time and money, and the repercussions of not doing stem cell characterization quite routinely can be felt all the way through the entire stem cell workflow. Now, with regards to stem cell workflows and kind of getting back onto this modeling course that we have here, stem cell workflows nowadays, especially as Omar alluded to when he was talking about neuronal cells and cardiomyocytes, they often extend beyond reprogramming or gene editing steps, right? When we're disease modeling specifically, we almost invariably differentiate those iPSCs into specific cell types. And again, we can do this, as you now know, with or without gene edits. And all of that allows us to better recapitulate disease-relevant phenotypes. Remember the cell modeling slide, selfishly, right? This is all a reciprocal system, and it all comes back to the human being, right? And that allows us to recapitulate these specific phenotypes of different diseases that we're going to be working with. Once we've generated our cells, right, whether it's a working bank of cells or a cryopreserved bank of cells, what we see a lot of nowadays, as I said, is folks differentiating those iPSCs into mature functional cell types. And as you might imagine, as with anything, right, there's a lot of different factors that govern this process of stem cell differentiation. But really at its core, I want you to think of this as a process that changes the cell to a more specialized cell type, right? We go from a switch, if you will, from sort of proliferative state to a specialized state. And this is driven by a combination of lots of different things. We've got genetic factors, we've got environmental cues, we've got signaling pathways. You can see much of that on the left here, all of which regulate gene expression. We've also got sort of an undue influence of the extracellular environment, right? We now know, modern understanding of stem cell biology, that microenvironment in which the stem cells reside really plays a big role in directing differentiation. Think of all the different physical and mechanical forces at play, right? We've got extracellular matrices, membrane potential, metabolic activity, cell-to-cell contacts, media systems, right? As we're talking about, all of this can greatly impact the differentiation process. At the end of the day, though, I think what that amounts to is quite remarkable, as you see here on the right. And again, we're not going to go into detail on this figure here, but it's a really nice visual representation of those endodermal, ectodermal, and mesodermal germinal lineages as they differentiate into the various cell types of the body. Now, at Thermo, we do have a number of different media systems and kits that support directed differentiation. Just as a quick example, we've got Essential 6 media. This is a growth factor-free base for differentiation. We also have ready-to-use kits, right, as you see here on the right, that help steer iPSCs into cell types of different lineages, like the definitive endoderm for cardiomyocytes, like neural stem cells. We can further differentiate those into hindbrain-like neurons and glia, and importantly for the context of our time today, midbrain foreplate progenitors and dopaminergic neurons, which we use to study Parkinson's disease. So in today's modern age, really the take home here is it's been made a lot easier. It used to be very, very much a lot of the work we did in the lab was through homebrew kits, sort of mad scientists mixing all these recipes together. But now companies like ours and a lot of the others out there offer ready to use kits, which makes it quite easy to use. Okay, on to Parkinson's disease. Any questions before we wrap up today with our case study?
There's been a bunch of questions in the chat, but I've been trying to get them as well.
Thanks, Omar. Anything else you'd like to add at this point before I do the case study?
No, I think you put it really well, Tom. And the one thing I'll say is I know Tom mentioned quite a bit of like the overall workflow. So if it seems like a lot, don't worry, right? We're going to be covering every single step in a lot more detail in the next following weeks. So you'll get a lot more detail. You'll be able to kind of understand how each step flows into the next as well. But I think, Tom, this is a great place to go into the case study, right? Just to show from start to finish what that looks like too.
Yep. And just to add to Omar's what Omar was saying there, please feel free to leverage the resources in this course, not only the content and the slides and the links and the data sheets and application notes that we'll share with you that we think are useful, but also us as well, the instructors. We want you guys to get a lot out of this. So if you have a question that pops up about something we've talked about in the course or something that's specific to the research that you're doing, we'd love to hear from you. This is a big part of what makes this course successful, is sort of the collaborative nature of the work. So don't be shy. Feel free to speak up, whether that's during the course or after the course as well. We always love to hear from you guys. Okay, Parkinson's disease. I'm sure most of you, or probably all of you, have heard of or perhaps even know someone that has Parkinson's disease. My dad has Parkinson's disease. It's quite a common neurodegenerative disorder that I would say first and foremost in Parkinson's is characterized by a host of motor symptoms, right? And by motor symptoms, I mean things like shaking, right? Bradykinesia or slowed movement, muscle rigidity, abnormal posture and pace when people are trying to walk or perform certain tasks. And what happens in Parkinson's disease is these motor symptoms begin quite gradually, but ultimately they get worse over time without fail, right? And they get worse and then sort of pile on, if you will, and include disturbances in things like behavior, emotions, sleep, memory, and so on and so forth. And at its core, what happens here is neurons or brain cells, right, in a certain area of the brain called the substantia nigra that we know is responsible for things like reward and movement. Those neurons in that area of the brain, the substantia nigra, they become impaired and die. And these are also the neurons that produce a substance called dopamine, which you may have heard of. And dopamine is something that plays a massive role in how we do things like think or plan or feel about a certain thing, right? Another thing with Parkinson's that is quite interesting is Parkinson's patients also often lose the nerve endings that produce a substance called norepinephrine, something you've probably heard of as well. And norepinephrine or norepi is a hormone that helps mobilize the brain and the body for action. Importantly, it does so, this mobilization, and therefore dictates how we react, right? Especially under stressful or anxiety-provoking situations. And so with Parkinson's disease, we've really got the loss of these dopaminergic neurons paired with the blunting, if you will, of those nerve endings, really is the causative agent or harbinger, if you will, that throws everything else out of whack here. And if you think about basic functions of the brain, right, things like sensation, perhaps perception, feelings, thoughts, behaviors, right, you can probably imagine how loss of those important dopaminergic neurons and the blunting of those nerve endings, right, could really be catastrophic, right? But in today's modern medicine age, right, how stem cell derived dopaminergic neurons might offer tremendous potential as a potential treatment for this disorder, right? And so there's lots and lots and lots of therapies that are sort of in the pipeline or in the works for stem cell derived dopaminergic neurons in the setting of Parkinson's disease. We've got some really, really cool researchers out there nowadays doing things like disease modeling, drug discovery, even functional engraftment of these dopaminergic neurons into the brain, right? And just from a very high level and basic kind of stem cell derivation workflow, it would typically look like something you'd see across the top here. I've put a couple of papers here at the bottom. They're, within the last 5 to 6 years or so. For the most part, I think they're really nice sort of overview of some of the work that stem cell researchers have done in this space, especially how it pertains to stem cells, the brain, and neuroplasticity, not just in Parkinson's disease, but also in other neurodegenerative disorders as well that we see throughout the lifespan, right? Which, as you could imagine, right, do benefit tremendously from things like stem cell therapy. And so lastly, as promised, so putting some of those principles today that we've talked about into practice, I wanted to share what I think is a really interesting case study or collaboration between the Parkinson's Institute in California and our team at Thermo Fisher Scientific. And so what we looked at in this particular example was the ability of stem cells to differentiate into those dopaminergic neurons, right? And we highlighted the importance of something here called LRRK2, LRRK2. LRRK2 is a pretty widely expressed protein that we know is a crucial factor to understanding the cause or the etiology of Parkinson's disease. There's a lot of data out there that links LRRK2 to many of the pathogenic mechanisms, if you will, that underlie Parkinson's disease. So think things like oxidative stress, mitochondrial dysfunction, the inflammatory response, right? So if you sort of think about things and take a step back, one potentially promising approach to maybe understanding and even treating Parkinson's would be to solve for this LRRK2 problem. So what we did in this case is we took a Cas9 expressing iPSC line, and we used it to genetically engineer a point mutation in this LRRK2 gene, specifically something called G2019S, which is a SNP conversion, single nucleotide polymorphism, that we know is associated with Parkinson's disease. We then took those iPSCs, we characterized them, right? Again, we want to do this routinely throughout the workflow, and then ultimately differentiated those cells into dopaminergic neurons. And then, as you see here over on the right, we performed a number of different cell-based assays to ultimately investigate the effects that this particular mutation had on the biology and function of those neurons. So after introducing that LRRK2 mutation, what we did first is confirm gene targeting by something called Sanger and next-gen sequencing, as you see here in the bottom left. As we talked about, we then ran a couple of different characterization assays. We did this to confirm that the edits didn't affect things like cell quality or influence stem cell identity. This included a couple of assays, one called the TACMAN scorecard to examine trilineage differentiation potential. You can see here that the parental mutant lines are quite similar, which is important. We also looked at something called the Pluritest and the Karyostat assays, which basically allowed us at that time to confirm that pluripotency and karyotype were unaffected by our genome editing process and line clonality, as we see over here on the right. Now, after we made those edits, we had specific control and mutant lines that were differentiated using that kit I talked about, our PSC dopaminergic neuron differentiation kit. What we found is that both lines differentiated normally. Also, both lines showed appropriate staining for a number of important neuronal markers like TH, or tyrosine hydroxylase here. TH is the rate-limiting enzyme in dopamine synthesis. It's a pretty widely used marker for dopaminergic neurons. We also looked at something here called beta-3 tubulin. Beta-3 tubulin is just about found exclusively in neurons, where we know it plays an important role in structural support and the transport of neurotransmitters. And you can see in these images here that the markers are comparable in the top panel, the mutant line, as well as the control panel across the bottom here. Now, in this assay here, we looked at something called neurites. And neurites are essentially neuronal processes, think of like fingertips, if you will, or projections from the cell body of a neuron. And what neurons do, quite fascinatingly, is they actually grow and tailor these different projections in response to different guidance cues that they get from their internal and external environment. And so what we did here to analyze that neuron biology and function is looked at neurite outgrowth using a live cell imaging system. And what we found here was a slower rate of neurite outgrowth in early time points for our mutant cells, as shown in the red box on the left, and then the top figure on the right here. But what ultimately happened is that neurite outgrowth actually stabilized over time, and the mutant cells actually grew longer neurites than both the parental control line and another control line that we had, something called alpha-synuclein mutants, which were also associated with Parkinson's and were used as an additional control. So we show here that in this particular LRRK2 mutant phenotype, we saw longer neurite outgrowth in those Parkinson's dopaminergic neurons. And at this stage in the experiment, that was likely as a compensatory or sort of adaptive panic response, if you will, in this mechanism to injury. Now, as I mentioned, one of the big hallmarks with Parkinson's disease is loss of those dopaminergic neurons. And so in order to further recapitulate elements of the disease here, we looked at the sensitivity of those differentiated cells to known toxins. In this case, something called 6-hydroxydopamine, which is a neurotoxin specifically taken up by dopaminergic neurons. In this case, we used our Cas-based high-content imaging assay and basically asked the big picture question of how the control or mutant dopaminergic neurons actually responded to this neurotoxin. And then we also used something called staurosporin in the middle here, which is a potent non-selective apoptotic agent, basically to set a baseline, if you will, for overall cell death in the control and the mutant cell lines. So we show representative images here on the left, and then quantified apoptosis or cell death data on the right here. In the panels on the right, you can see quantified data from the imaging assay, and you can see in this LRRK2 mutant box here in the second column that they're much more sensitive to neurotoxin exposure and showed increased apoptosis. And we also showed, as I mentioned initially, that stress can worsen symptoms in patients with Parkinson's disease. And so in order to mimic stressful conditions here, what we did is repeated this same experiment, right, in the absence of glucose, as we're looking at in this bottom graph here. And again, the LRRK2 mutant line showed increased apoptosis compared to the parental line, again, highlighting that Parkinson's dopaminergic neurons are more sensitive to neurotoxins. And this is something that's in agreement with the literature and importantly mimics what you'd expect to see in a patient with Parkinson's disease. And then last but not least here, we also know that this particular mutation, as important as it is, I said, impacts expression of a number of other genes as well in dopaminergic neurons. So we used a gene assay here called the TACMAN and found that by introducing this particular mutation, we saw an increase in expression of other LRRK-regulated genes associated with neurodegeneration as compared to the control panel and the control lines here, as you see on the left. As we're looking at on the right here, we saw no changes in gene expression in our other control disease line, our alpha-synuclein mutant line, which again is consistent with the literature and really, I think, highlights not only the complexity of the disease here, right, but also that changes in expression of those genes is not only specific to this LRRK2 mutation, right, all of which is important in the induction of these Parkinson's-associated phenotypes. So just to summarize quickly this case study, we took these LRRK2 mutants, we generated them using a Cas9 expressing iPSC line. Those edited iPSCs were shown to maintain pluripotency and a normal karyotype after genome editing. We did that using a number of different characterization assays. We then differentiated those cells into midbrain dopaminergic neurons, in which this specific mutation, this LRRK2 mutation, as I said, impacted neurite outgrowth, neurotoxin sensitivity, as well as gene expression in those cells. So again, I think this is a really interesting study and one that has a lot of clinically relevant implications. Okay, that was a lot of information, I know, for our first talk together today. So I'm going to leave you with this hopefully nice, calm overview slide. We touched a little bit on biosafety in the beginning before diving into a couple of cell culture basics and then diving a little bit deeper into the stem cell disease modeling workflow, where we emphasize what we can do in modern technology, right, to leverage stem cells to help us create these physiologically relevant models by doing things like reprogramming somatic cells, right, blood and skin, for instance, into iPSCs. After that, we can then genetically engineer those cells through technologies like CRISPR or TALENs. It's important that we characterize and qualify those cells along the way, right? And then ultimately, if we're happy with those cells, nowadays we can differentiate them into physiologically relevant functional cells, right? Like dopaminergic neurons that, again, mimic a lot of the different elements of human biology and allow us to have really useful models to study disorders like Parkinson's disease, which we talked about today. I will stop there in the interest of time. Couple of things. First and foremost, we're so glad you joined the course. Honestly, we mean that. We had a big turnout and a lot of success with the course last year, so we're glad you're part of it. Appreciate your attention and, of course, your interest. I know that we'd all be happy to take any questions either now or offline. As I said, I'll put this slide here at the end of each talk with a couple of different links at the bottom, as well as a few different resources that we'd like to share. You'll receive this information within a day or two after each of the subsequent lectures. There is a wealth of materials out there, not just on the Thermo Fisher website, but in the World Wide Web. There's loads and loads of content on stem cell biology. So if you feel like digging deeper on any of this, please have fun with it. And all of this, I think, is really important and useful information to have in your back pocket moving forward. I put this slide up here too. It reminded me of Easter yesterday a little bit. Hope those that celebrate had a nice Easter. But there's always one annoying piece left over. So hopefully we figure all that out as we progress with the course. But thanks for your time. Happy to take any additional questions.
Thank you, Tom. That was awesome. There's maybe just one more question in the chat around.
Go for it, Omar. Yep.
Can you hear me?
Yeah.
Yep.
Yeah, I was to say there's one more in the chat around issues that cause domino effects down the line when using like kits versus like home brews or like basal media growth factors, right? Like the question is, how is the reproducibility defined when using these kits specifically compared to the home brews? I don't know, Tom, if you want to comment, I can also
no, feel free, Omar. Yeah.
Yeah, just to comment on that, I think this is a great question, right? But and the shortest answer I could say is yes, there can be downstream effects that are caused by changes due to reproducibility issues upstream when it comes to either the kit or the protocol you're using. That said, it's not always the case that kits are better than home brews, right? It's more about that protocol itself, and the cell type that you're trying to generate. If that protocol is robust, you will still have very, very good results downstream when you differentiate. The benefit with the kits is that there is an onus on the supplier of that kit to ensure reproducibility as well, right? So with the kits, there's maybe one more added measure that you are able to kind of rely on a little bit, that you're getting what, you're always getting what you're buying. And if not, excuse me, you can go back to that supplier and say, hey, there's something going on and, something is off. My cells aren't behaving the way that they should be. But that's not to say that it's, better in a way than home brews or protocols where you're adding the media and the growth factors. Sometimes it's a little bit easier, but that also comes at a cost of potentially not knowing what you're putting or adding into the cells that the company is supplying for you in, formulation media A or formulation media B, right? That you don't know what's exactly inside of them. So again, it's a little bit of a toss-up, Jeanette, but typically it will depend on the protocol and on the cell type that you're trying to derive.
Great question.
Yeah, good, very good question.
And much of what Omar and I do day to day is supporting, and giving talks and sharing the knowledge on a lot of this stuff. And I think, coming from again, neurobiology background, which is at the best of times a very finicky space to work in. There's a lot of old school protocols that might have worked for someone a long time ago that I sure as heck couldn't get to work in the lab on some of my experience. And I think, again, we are from Thermo Fisher. We're not necessarily just advocating for us, but I think when you take out some of the uncertainty of home brew and you go to an established company where they've got tried and true data to show some of that reproducibility. They've got teams of scientists that are there to support. And nowadays as well, they can also be cost efficient to buy kits versus trying to pull together a tiny bit of this and a tiny bit of that and a tiny bit of this, also quite complicated home brews. But again, that's not to say one's right and one's wrong, but certainly to each his own. And as I said in the beginning, a lot of stem cell biology researchers and neurobiology researchers are quite finicky and old school in their approach. So you may be in a lab where you're stuck using a particular method, and whether that's a home brew or an established kit. And as you get further along in your research, you might want to try the other and try something new.
Yeah, good point. One more question, Tom, a couple more actually, but from Anthony. So with the advancement of iPSC technologies, do you foresee the future is allogenic off-the-shelf therapies? How close are we? Another really good question.
It's a great question. Yeah.
Yeah. I would say the goal would be yes, like an off-the-shelf allogenic therapy that, you can, you don't have to necessarily wait for, that is readily available, that is potentially cheap as well. We are super close, as I mentioned. We do have now two stem cell approved therapies. Both are in Japan, but I think as a proof of concept, there's some really good clinical data coming out about the efficacy of those types of therapies. Most of them in the beginning are going to be autologous because that's an uh, more of a personalized approach, which might be there's a point to make here that this could potentially be a little bit safer in the beginning, right, as well. But I would see allogenic as the future, but not the only future, right? Because
Yeah.
autologous therapies are very, very important. They're very precise and tailored to the individual. There's some research to also show that personalizing the treatment might also benefit outcome to a certain degree. So people are still investigating that. But ultimately, I would say it would be great if you could just go, pick up the treatment and not have to wait for its manufacturing with autologous.
Yeah, in general, it's just there is a immune privilege region, something like brain, eye, or that area, but still, there is a immunological issue. Also, another technology people raise up is like a universal iPS. Those
Mhm.
one is like more evade the host immune system. So that is another research area is people is doing.
It's so interesting that you mentioned that key type because they're using it in so many modalities as well. There's iPSCs that are being used as potentially or being studied for cancer vaccine as a universal cancer vaccine because
Exactly.
a lot of the cell surface markers that are present on pluripotent stem cells are also present on cancer cells as well, right? Because they're very proliferative, they have somewhat similar properties. And there's scientists that are trying to figure out, well, can we use stem cells as a vaccine for cancer, right? And there's some good data on that too. So there's so much interesting things happening here. Awesome. Um, I don't know how we're doing for time, but maybe one more question. Um, is it necessary to characterize differentiated cells every time if you're following the same protocol? Another really good question. You want to take this? I could take this too.
Oh, yeah, no, yeah. I mean,
Yeah,
again, I think that's certainly to each his own. I think it's important to characterize as best you can, as often as you can. There obviously are a lot of limitations in that, usually money and resources and time being a few of the biggest variables. Um, Again, I think at the end of the day, it comes down to what resources you have in the lab and the big question that you're going to be answering. If that question and answer involves functionality of the cells, I think when we start to get away from differentiating ourselves at any point in the workflow, the downstream costs of that might be big, big changes in data consistency or functionality. And so, yes, I would say characterize as early and as often as your resources allow. But Omar, curious to hear what you think on that.
no, I completely agree. And also, it would depend on what you really mean by characterize, right? So, characterize, if you're referring it to in a sense of I need to assay the cells that are kind of coming out of the pipeline at the end of my differentiation, I would say always yes, right? You always have to do some sort of verification that you are getting the cell type that you want, even if it's a protocol that you've done 100, 1,000 times, right? There's always that risk of, hey, this didn't work or the differentiation didn't work. So verifying that you're getting the correct induction is important. The second part of that, or the other flip side, if you're using characterize in a similar way to what Tom was talking about characterizing like the genetic profile or the status of the cells, that may not be as important after differentiation unless you are using the cells in some sort of therapy, right? Where they're being transplanted back, then you want to make sure that you're not adding anything in that is that had genetic material alterations or some proto-oncogenic gene that can mess these cells up after you transplant them. And because these cells, come from a source that typically can proliferate quite readily, right? Pluripotent stem cells can proliferate very readily. We don't want to introduce that risk when we're doing grafts or transplants back, right? So to your point, I think it's Tom. To your point, I think it does depend on what you mean, but it is important to make sure you're understanding what you're getting out of your differentiation every time. But uh, past that, the safety profile is usually related to cell therapy applications.
Yeah, just I want to add one more thing is quality control is one of the big part of the stem cell research. That is Oma is explained very good way. And then also another one is standardization and the reproducibility. That is the reason why our center with a collaboration with the Thermo Fisher automation team, and we established the robotic automation to generate iPS cell, gene editing, as well as tissue differentiation. Those kind of thing is kind of another effort to minimize the variability. And then those kind of activity will continue in the stem cell field.
Yes, absolutely. And a good call out here, Kitai, because specifically also for this group, right? Stem cell culture in general is, I mean, I don't want to make the comparison, but I would say it's several notches higher in complexity than traditional, cell culture. There's a lot of things that we have to consider. There's a lot of things that we pay attention to. We have to, be as gentle on these cells as possible because, as Tom mentioned, they have the capacity to pretty much turn into everything in your body if given the right signals, right? So to Kitai's point, you have to make sure that your process is controlled, right? That every time you differentiate or you culture these cells, that you're paying attention to these little details that can get them to, change course one way or another, right? And in that sense, the reproducibility becomes a big, a big important factor. I know one thing that we're doing at Thermo as well is trying to see how, even like experienced operators, how they differ, right? When we test things internally, we make sure that, if a product protocol is developed by one person, that it has to be reproducible in the hands of at least two or three more other people that are just as experienced, maybe in different sites or in different places. And that's to speak again to just that reproducibility issue. So adding robotics in, automation tries to eliminate all of that so that you can get to that higher standard too. Sweet. I know that there's a little bit more questions here, but maybe some of them are about like work experience and things like that. But Kitai, what are you thinking? Do we have some time to answer some of them, or do we leave them for
Uh,
perhaps for the next call?
yeah, it's depending. We have a couple of minutes left, so therefore either you can mention or we can discuss further next week.
Okay, let's see. Let's see if I can get like a general sense of what they're talking about. And there's, you see editing small molecules, uh, recapitulating. Okay, so there's a good one in here about limitations of iPSC-based models in accurately recapitulating late onset disease like Parkinson's. Right? This is a great question, and something we're going to mention when we talk about the differentiation portion of these cells, right? During development in our bodies, it takes us 9 months to go from, the embryo to a, a tiny baby at birth, right? But then 18 years later, perhaps even 21 after, to become fully developed, right? Like brain, full body, everything is sort of like mature. And squeezing that into an in vitro protocol, you don't want to wait 21 years before you have data, right? That's just crazy. So there are certain ways in which we can speed up this timeline or this process in the lab by recapitulating those, you know, routes that these cells take and forcing the expression of certain factors or genes or pathways, right? That being said, Pooja, what you mentioned was is probably one of the biggest hurdles that we face in the field of making sure that our stem cell models are mature enough to recapitulate disease. And like I said, there's a bunch of different techniques or mechanisms that people are discovering or that people can employ, I should say, to focus a little bit more on maturity or mature models. So using organoids has shown to be a little bit more or representing a more mature population of cells that are iPSC derived in general, not always, but in general. And if you want to expand on that, there's a little bit of work where you can take these organoids and almost fuse them together to make assembloids that have even been shown on, like the hepatic and liver side to increase maturity. Uh, but there's also really interesting things that you can do. And I'm thinking about this just because of Artemis's recent launch, right? But if any of you have heard of some of the stem cell work that's happening on the International Space Station, right? Researchers send up their cells and their organoids, mainly brain organoids that I've seen, in order to age them in space in zero gravity. And what they found out was it was something crazy. Like a month in space is equal to 10 years in like on Earth when it comes to the maturity level of these cells. Something is happening to them at zero gravity that's causing them to mature a little bit faster somehow. So there are things that you can employ, but we're still working on that a little bit. Sorry, this was a long-winded answer, but I thought it was super interesting, so I think it. With that, maybe we can finalize today's call, and Tom, maybe you and I can just recap next week. Did Tom drop? He may
Oh,
have.
I can recap.
Ah, yeah, that's okay. I can do the same as well,
Yeah,
Kita. Yeah.
yeah.
Yeah, but next week, there he is. You
Sorry.
just got disconnected. Yeah, so next week we're gonna dive like I said, a little bit deeper into the start of that workflow that Tom was mentioning around, the reprogramming. And we'll continue our discussions. I'm hoping that, today was a good start for everybody. Thank you, Tom. I think that was a really great introduction. And Kitai, anything from you to kind of close us out?
Yeah. Especially as Omar said and Thomas said, is a stem cell research field in progression. So therefore, for example, something like an aging issue, how we can incorporate aging, that is another big issue a number of researchers is studying. Another question about CRISPR versus chemical, how we can mimic it, and then there is a technical advantage if you use the chemical. That is also another big study field. And then regardless, we will keep going on in the next lecture. And thanks for Tom and Oma for another excellent lecture as they did last year. And I also would like to express my appreciation to Nicole DeJesus and senior regenerative medicine specialist from Thermo Fisher Scientific, and for coordinating all of the administrative support. If you have any question regarding specific product, feel free to reach out to Nicole. And then I also thanks to James Kim and Javier Fernandez and the administrative manager of the UCLA Human Stem Cell and the Genome Engineering Center for their support. All lecture content will be uploaded into the center YouTube channel after completion of the course, and all of the course material will be distributed after this course. And then next week, as mentioned, we will cover reprogramming to human iPSC, including detailed mechanism, key material, and chemical kinetics. While some of the component is based on the product from the Thermo Fisher Scientific, but similar product is also available from other resources. So we will also discuss broadly applicable product using the across different platforms. And this session will be valuable even for those with prior experience, as detailed chemical kinetics are not typically covered in the standard protocol. Thank you very much, and see you next week.
Thanks, everyone.
Thanks,
Bye.
everybody. Have a good one.
Yep.